Good afternoon, everyone. Thank you for joining us today. My name is Marissa Waterman, and I'm the Marketing Director here at American Academy of Environmental Engineers and Scientists. We're so excited today to be doing this webinar with Dr. Lauren Stadler. She's Assistant Professor with Department of Civil and Environmental Engineering at Rice University. Her research is focused on advancing sustainable and safe wastewater management systems, and she works closely with the City of Houston's Water Utility and Health Department. Today, Dr. Stadler will be discussing the surveillance of Houston's wastewater to track community COVID infection. Before we get started, I just want to mention that you'll be able to message your questions at the end of the presentation. Once the interview is over, you'll be able to type your question into the attendee chat. You'll see it on the left-hand side of your screen. And then we'll also share Dr. Stadler's email address, so you can email her directly with any additional questions you may have. Okay, let's get started. Good morning or good afternoon, Dr. Stadler. Thank you for joining us today. We're ready when you are.
Thank you so much for the introduction. I'm going to share my screen here. Okay, so thanks again for the introduction. I'm really pleased to be able to share some of the work that we've been doing in close collaboration with the City of Houston. And this is really an effort that's been led up by the Houston Health Department and also in collaboration with Houston Public Works. So I'm going to turn off my video just so you can see the screen in full view. There we go. Okay, so just before I get started, I want to give a shout out to my research group here at Rice University. A lot of the work that I'll talk about today is the result of their hard efforts and their long hours spent in the lab, and they're a great group of students to work with. And this project has really brought our lab together in many ways, because when the pandemic hit, I was, it started out with, let's get some wastewater and see if we can detect SARS CoV-2 in it. And everybody was on board to help out. And then it's expanded to this really large project that many, many people in my lab have contributed to at this point. And then aside from wastewater-based epidemiology, my group works on many different projects related to biological wastewater treatment and environmental microbiology. And we use tools from synthetic biology to study environmental phenomenon. And we work on problems such as how to improve the resilience of wastewater treatment systems, how to convert waste into valuable products, and how to mitigate the spread of antibiotic resistance. But today I'm talking about our wastewater monitoring project. And first I need to just give a shout out to the many people that are involved in this project that allow it to function on a really a daily and weekly basis. So this has been a huge collaborative effort that has involved and really been led up by Lauren Hopkins at the Health Department and her group there, but also Houston Public Works, which has allowed us to access wastewater samples all across the city, and I'll get into that. And then the analytical team has been a partnership between the Taylor Lab at Baylor College of Medicine and my group at Rice University. And then the statistical modeling has been led up by Kathy Enzer, and the hydraulic modeling by Phil Bedient. And this started out really as a rapid project funded by the National Science Foundation in collaboration with researchers from NC State, Howard University, and USC. So here's an overview of what I'll talk about today. First, I'll just kind of give a background about what wastewater-based epidemiology is. Then I'll talk about some of the methods that we spend a lot of time evaluating to understand how to best, at least within our lab, measure and quantify SARS CoV-2 concentrations in wastewater. And then I'll talk about the wastewater surveillance system that's been implemented in Houston and how that data is used by the health department on a weekly basis. And then I'll end by talking about some additional studies we've done to try and understand the variables that impact the viral signal of, in our wastewater system. So first, as an overview, a large portion of infected persons, both asymptomatic and symptomatic, shed virus in their stool. And that virus enters the sewer net. Oops, this is advancing without me wanting it to. So give me 1 second. I'm just going to make sure I turn off the timings. There we go. Um, 1 minute. I think I have to share screen again. Here we go. Okay, great. Thanks. Apologies for that. Okay, so that virus that's excreted in the stool enters the sewer network, and where that wastewater is then transported right to our through our network to our wastewater treatment plants. So wastewater thus represents this pooled community sample of everyone who goes to the bathroom and contributes to that wastewater stream. Um, and that allows us to basically sample the wastewater treatment plants all across Houston and essentially get a snapshot of the entire population of Houston in terms of the levels of SARS-CoV-2 that are present in the wastewater. We take that wastewater sample and we analyze it for the concentration of SARS-CoV-2, the virus that causes COVID-19. And these results, and many groups around the world have been doing this, and these results can either be qualitative, right? So just presence, absence, is the virus there, is it not? Or better yet, they can be quantitative, which can inform how much of the virus is present in the sample and whether that quantity is increasing or decreasing over time and across space. And then increasingly, researchers, including our group, have been spending a lot of time thinking about what additional information we can get from wastewater. So actually sequencing the viral genomes present in wastewater to try and understand which variants are circulating in our communities and potentially even identify emerging variants in our communities. There we go. So there's a number of advantages of using wastewater monitoring for tracking disease and for disease surveillance, which is why so many groups across the country and all over the world have really adopted this approach in response to the COVID-19 pandemic. First, diagnostic tests are likely only picking up a fraction of the total SARS-CoV-2 infections. Many people are asymptomatic or only mildly symptomatic, and these people might not go get tested, which doesn't mean that they're not spreading the virus. And even if you do get tested, there's often a lag time between when you might first start experiencing symptoms, right? So infection onset, and when you actually go get tested. And at the beginning of the pandemic, there was also a pretty significant lag time in terms of when you got your results back from your test. Which all of this stuff is precious time if we're thinking about disease spread, because you may not change your behavior, which could lead to the spread, you interacting with people and infecting others. Wastewater monitoring represents a significantly less expensive way to do mass surveys than individual testing. If we think about in Houston, even at peak testing levels, we're doing in the thousands of individual PCR testing. With the wastewater monitoring work, we're essentially covering, 2 to 3 million people every week by grabbing a sample of their wastewater. Wastewater allows you to get coverage in areas where there's little clinical testing information. And so there are many communities in Houston where testing penetration rates are not super high. And so we can use the wastewater as kind of the primary insight into whether infections seem to be increasing or decreasing in those communities. And kind of alongside that, you can implement in a way that allows you to identify specific geographic areas of concern. So you can monitor at the wastewater treatment plants, but increasingly groups have also been monitoring within the sewer shed to get more resolved information about which communities are experiencing community spread. So this is not an entirely new approach, right? People have been using wastewater-based epidemiology to do disease monitoring for some time. And there's a lot of literature that has demonstrated for the use of other viruses. So for example, poliovirus, but also some other enteric viruses like adenovirus, norovirus, rotavirus. It's also been used to look at illicit drug use like opioids. So basically monitoring for the presence of these drugs in the wastewater. These previous studies really demonstrated this approach and its potential to be used for early warning of outbreaks. And also, in the case of poliovirus, the idea was that this could be used to basically confirm the absence of virus circulation in a population. And then if a virus was detected in the wastewater, it would basically trigger a vaccination effort in that community. And so, of course, when the pandemic hit, there were a huge amount of interest immediately in basically grabbing wastewater samples to see if we could also see SARS-CoV-2 present in the wastewater sample. I think one of the first studies that really got huge headlines was University of Arizona that used this approach to basically isolate 2 asymptomatic students in a dormitory. All the students had to be tested before they moved back into the dorms and test negative, but 2 students had contracted the virus between when they tested negative and when they actually moved in. And the wastewater monitoring approach basically allowed the researchers to identify this dorm and then do individual testing of everybody who had moved in and then find these 2 asymptomatic students and quarantine them before it resulted in an outbreak. And then increasingly, researchers are showing that you can use wastewater sequencing, so sequencing of the virus to actually identify variants of concern, especially these highly transmissible variants like the UK variant, South African, and Brazil, circulating in communities before clinical testing reveals the presence of these variants in the communities. And then there's been a number of stories that have come out of Houston in particular because of these efforts in which they've been using the wastewater data to get a picture of where things are improving across the city and where things are getting worse, and therefore resources need to be directed. And oh gosh, this is not updated at all. I meant to do this before this. So this is an old slide, but this is the great work of the folks at UC Merced who've been managing this dashboard, or really it's a compilation of all the groups and dashboards and universities and countries across the world that have implemented wastewater monitoring for SARS-CoV-2. And these numbers are very outdated, I think. So these numbers are actually quite a bit higher now, but it's kind of amazing to be part of this incredible community of researchers who are all working and sharing information and sharing protocols and best practices in real time to implement wastewater surveillance of SARS-CoV-2. And so I've learned a lot from this community, and I think all of our science is better for this because there's so many people actively working in this space and working in real time in response to this pandemic. Okay, so now I'm going to talk about methods that are used to detect SARS-CoV-2 in wastewater. And I'll talk a little bit about the studies that we've done to evaluate the methods that we're using for SARS-CoV-2 detection. So first, I'm just going to show here this figure on the right is the structure of SARS-CoV-2. It is an envelope virus, and that envelope protects the genetic material inside it. And the way we actually detect SARS-CoV-2, both in clinical samples and in wastewater samples, is actually by detecting just a tiny fragment of that viral genome. We detect these very short fragments of this RNA genome, and these fragments are referred to as N1 and N2. These are genes. These genes are commonly used in diagnostics as well for clinical testing, and also there are other fragments you can use to detect SARS-CoV-2, but we're focusing, and many other groups have focused on using the N1 and N2 genes for the detection of SARS-CoV-2. And what's important to know is because we're only detecting these very short fragments, the virus does not need to be intact for us to be able to detect it. Just the fragments of the genome need to be present. And this is important because we actually don't know a whole lot about the form of the virus in wastewater. And there's a lot of evidence now emerging that actually the dominant form of the virus in the wastewater is actually a degraded and non-intact form. But I don't think that's particularly critical. That's important if you think about the potential of exposure. So, whether there's infectious virus in wastewater, but really the goal of our study here is to actually just measure the amount of SARS-CoV-2 that's present in the wastewater to get a sense of how much is in the community. And so it's not important that the virus is intact. It's just important that we're able to detect these fragments. And the reason we spent quite a bit of time, and many groups have spent quite a bit of time evaluating methods for detecting SARS-CoV-2 is because wastewater is it's a complex matrix, and the virus is present at extremely dilute concentrations in the wastewater. Um, and it's also full of other things, right? Like organics, solids, toilet paper, other bacteria, other viruses that can potentially interfere with the ability to detect this dilute signal of RNA from the virus. So the first step associated with the quantification of SARS-CoV-2 in wastewater is a concentration-based step. And so this is really to get it to concentrate it because it is so dilute in the wastewater, and to get it at higher concentrations so that it's easily quantifiable and that you have a sensitive limit detection limit in your assay. So we've spent a significant amount of time developing and demonstrating different methods for detecting SARS-CoV-2 in wastewater. And there's, like I said, there's the first step is concentration. And then after you do that concentration step, there is an RNA extraction and purification step. And then after that is the quantification step. We actually measure, the number of the fragments of this viral genome present in the sample. And so this is just a schematic that shows all the different things that we've evaluated in terms of head-to-head comparisons in our method development. And we spent the most time focusing on the concentration step because it ends up being the most critical step for having a sensitive and low limit of detection for the detection of SARS-CoV-2 in wastewater samples. So I'll talk about a study we did where we compared these 5 different concentration methods. And then I'll just briefly talk about the RNA extraction comparison and the quantification comparison that we did as well to end up with the final method that we use in our lab for quantification. So, like I said, we compared 5 different concentration methods, and we picked these 5 different methods because these were all methods that we saw were being used by many different groups all across the country and world working in this area to measure SARS-CoV-2 in wastewater. And we did this on 6 different wastewater samples. So we got wastewater from 6 different wastewater treatment plants and then did a head-to-head comparison of these 5 different methods. And we compared them both in terms of the amount of virus they recovered, but also in terms of practicality. So things like cost and throughput. And this was very important for us because we were basically spinning up to be able to do, hundreds of samples per week. And so cost and throughput were really critical in being able to operationalize this for the health department here in Houston. And this study really gave us confidence that we're using the best practices that would allow us to generate, actionable and reproducible data from our analysis. So these 5 different methods all kind of vary a bit in terms of the mechanism by which they concentrate the virus. So the first 2 use electronegative filtration, where you actually filter the sample through a filter and absorb the virus to this filter. And the difference between the first 2 methods is just how we then remove the virus from the filter after we absorb it. So A uses bead beating, where we physically beat the filter and remove the virus that way. And then B use an elution-based approach, where we actually push a solution through the filter to basically release the virus. The next method, C, ultrafiltration, uses size exclusion to concentrate the virus. And then D, PEG, is a precipitation-based method where you precipitate the virus and the nucleic acids. And then we compared that to a direct extraction method where we didn't do any concentration. So we just directly took a very small amount of wastewater and extracted the viral nucleic acid from that. So here I'm showing really the most important results summary from this study. We also spiked all of our samples with a surrogate virus and quantified that as well. But here I'm just showing the results of the quantification of the SARS-CoV-2 in these 6 different wastewater samples, which are shown on the x-axis. So each one of these letters represents a different wastewater. And then the different methods grouped on the x-axis. And so again, the goal is to evaluate the recovery and the sensitivity of these methods to decide which one to use for routine monitoring. And what we see here is that direct extraction had the highest virus yield for the wastewater samples. And this makes sense because there's no concentration of the virus involved, and all concentration processes have some losses associated with them. And so that's why you see the highest yield with direct extraction. And then after direct, the HA filtration methods, so really the electronegative adsorption methods, had the next highest yield, followed by PEG and ultrafiltration. But it's important to note that yield and recovery is only one factor that you need to keep in mind when you're deciding on which method to use. So here in green, I'm showing the limit of quantification. So each of these methods has a slightly different input volume. So starting amount of wastewater that we input into the process. So with direct extraction, we started with only 300 microliters of wastewater. Whereas with HA filtration methods, we started with 50 milliliters of wastewater. And so because of these different input volumes, there's actually very different limits of detection. And so when you overlay the limits of detection or limits of quantification on here, what you can see is that while direct extraction had the highest yield of virus, all of them, it was above the LOD in that we detected something was present, but it was below the limit of quantification, meaning it was at such low levels in these samples that we couldn't confidently get a quantitative number to basically be able to do trend analysis week over week, because we're so close to that limit of detection and really between the LOD and the LOQ. And so with these two pieces of information, what we can see is that we wanted to pick the method that both had balanced the yield of the virus and being the furthest away from that limit of quantification. And so the method that we ended up using and we use currently, is HA filtration plus bead beating, because it both has relatively high yields, but it also puts us by far the furthest away from our limit of quantification, which allows us to do quantitative analyses week over week to compare virus amounts or concentrations in the wastewater samples. And then in terms of practicality, we compare them in terms of cost, startup costs, consumable costs, and throughput. And again, kind of the main takeaway here, I'm not going to go into a lot of the details here, but the main takeaway was that HA filtration plus bead beating had the best balance of lowest cost, highest throughput, and furthest from the limit of quantification of all the methods. And so this is where we ended. This is the method that now we are currently using. And this has actually been published now, accepted for publication at Water Research. So in addition to comparing concentration methods, we also compared RNA extraction methods. We've actually compared an additional kit on top of this now, but we initially started by working with QIAGEN kits and comparing it to Maxwell kits. And we actually switched over for very, very practical reasons, in that QIAGEN had a huge shortage of reagents and supplies in the middle of the pandemic, I think because everyone was trying to get a hold of these kits for viral extraction. So we shifted over to Maxwell, but that actually turned out to be, when we did the head-to-head comparison, overall, the Maxwell kit performed better in terms of yields of virus from the same sample. So these are basically, we just extracted the exact same samples through the two different methods. And you can see in general, the Maxwell dots in red are higher than the blue dots. And so we felt confident in being able to switch over without impacting the sensitivity of our method. And then finally, we compared quantification methods. So our partners at Baylor use reverse transcription quantitative PCR, and we use digital droplet PCR. And these, I'm not going to go into the details of how these exactly work, but they vary a little bit in terms of kind of how the actual quantification of these targets occurs. What we see is that there's a really strong correlation between the results generated by qPCR and ddPCR. So you will get consistent findings whether you use qPCR or ddPCR, which is good because our partners at Baylor are using one method and we're using another. But in general, we see that ddPCR actually is more sensitive than qPCR. And so here's again just looking at several of the same extracts that we quantified via the two different methods, and we see that ddPCR yields higher copies of this viral genome per RNA extract. So this pushed us towards using ddPCR for our quantification in our lab. And then finally, like I said, wastewater is full of potential inhibitors that could that have the potential basically to interfere with your ability to detect these small fragments of RNA in your sample. And so in order to ensure that we weren't experiencing significant inhibition in our samples, we did an experiment where we took wastewater samples and those extracts and spiked them with a known amount of a synthetic SARS CoV-2 RNA. And then we basically compared the spiked to the known amount spiked to an unspiked sample to assess the degree of inhibition, whether we basically got back what we expected in terms of what we spiked in. And so no inhibition would be like we get back everything we spiked in, the fraction of spike recovered. And what you can see, at least for all of these samples, is that we saw very close to no inhibition in all of the samples that we evaluated. And so we felt confident with our method using ddPCR that we were not having significant inhibition occurring in our analysis. And so our concentration, extraction, purification, and quantification protocols were sufficient for purifying this RNA before detection. And so just to kind of summarize where we're at now, we ended up with this electronegative filtration method with the bead beating. We actually are now using a different RNA extraction method called chemagic, and we switched over to this largely because we needed higher throughput, and I'll talk about that in just 1 minute. But basically, we've expanded the monitoring across Houston quite a bit to the point where we needed something that was just a higher throughput system, and that's how we ended up at the chemagic. And then we're doing quantification of this virus using digital droplet PCR. And again, the groups all over are using different methods. And I don't think there's one right method, right, that to use here for doing this. It's more about understanding and really characterizing the limitations and the benefits of the method that you do employ. So you have a very well characterized limit of detection, that you're able to process the number of samples you receive a week. So the throughput was a huge issue for us, and also that it's performed in a timely manner and in a way that's within budget, because this also can get to be quite expensive when you start to do hundreds of samples a week. Okay, so now I'll shift to talking about the data that's generated and how it's used. So here I'm showing a map of Houston. And there are 39 wastewater treatment plants across Houston, which is kind of unique for a major city. I think most major large cities have just a handful of more centralized treatment facilities. Houston has 39 wastewater treatment facilities that range in size from serving 5,000 people to over 500,000 people. And while Houston Water, I think actually doesn't like the fact that there's 39 treatment plants, because that's a lot of treatment plants that they have to manage and maintain and staff. In this case, it really was an advantage because it gave us both spatial, basically we have a spatially, we can compare both over time within a sewer shed, but we also can compare over space by looking at the concentration of SARS-CoV-2 within each of these 39 treatment plants and comparing them to one another. So because of the success of this initial wastewater treatment plant sampling effort, this monitoring has expanded quite a bit. And so now we're also, the health department has implemented or put in these manhole monitoring composite samplers at 51 schools in high positivity rate areas, at 23 and counting congregate living facilities. So these are nursing homes, jails, homeless shelters. And then in addition to that, some of these wastewater treatment plant service areas are really large. So like 69th Street is by far the biggest. And so they've started to break up 69th Street into smaller sub-sewer sheds by putting in samplers at lift stations. And the plan is to potentially increase the number of lift station samplers to close to 100, so you can get near zip code level information about virus levels in communities. And so this is just a plot showing kind of the location. And I think these are almost all the school samples across the city where there are manhole samples near these schools in high positivity rate areas. So here's an overview of kind of what happens every single week. These samplers are 24-hour composite samplers that grab untreated wastewater. And every week we get one sample from each of these locations. Those samples are collected by Houston Water and Houston Health. They transport those samples to our lab at Rice or a subset of them to Baylor, where we each quantify in replicate the concentration of SARS CoV-2 in the samples. It was really important to the health department to have two different labs initially independently analyzing the wastewater samples so that because especially as we were spinning up these methods, a lot of this was under development. We hadn't done this extensive method study. And so we wanted to have two groups to basically verify and check our results against each other so that they felt confident in using the information to actually take action and direct resources. The data that is generated is input into a statistical model that's used to identify geographic areas of concern with significant increases or decreases in virus concentration. And that information is then translated to healthcare professionals and strike teams to then prioritize specific zip codes for interventions. And these are interventions such as additional testing units, increasingly now additional vaccination clinics, but also just messaging and education. And I'll talk a little bit more about how the data is used as well. So first I'll talk, before I talk about the manhole data, I'll talk about the wastewater treatment plant data and how that's used. So here on the right, I'm showing a figure that's showing the wastewater viral load from 16 wastewater treatment plants that we've been monitoring by far the longest. We started out just with 16 and then expanded to the full 39. And what you can see, so the viral load is shown in green, and the 95% confidence interval in the gray band around it. And then the positivity rate, so basically number of positive cases from clinical data over the total number of tests performed, is shown in the blue. Each actual daily measurement's the cross, and then there's a 14-day moving average shown in the dark blue, and then the spline or the fitted data is the other curve. But what you can see is there's a really strong correlation between the wastewater viral load and the positivity rate. And this was really critical for us to demonstrate because it gave the health department confidence in being able to use this data basically to monitor within the sewer sheds as well, not just across the whole city, and use the wastewater as a kind of a proxy for infection dynamics within communities. And this was also particularly important because there's some areas where there's really low testing data available. So there's just the positivity rate data is sparse and highly variable. And so in those areas, the wastewater data is really the primary means by which the health department has to monitor community infection dynamics. Okay, so like I said, all the data that's generated from these analyses is input to a statistical model. This model accounts for the differences across two different labs. And we spent a lot of time thinking about how to communicate these viral load levels to health department officials so that it was understandable and useful. So actually the way we present it now is we present it as a level as a percent of the July 6th viral load. So the July 6th in Houston represents when we had our peak positivity rate in Houston. As you can see here, it was a bad time. And so I think this helped. So we report it as a percent of this because people kind of have this as a baseline for how bad things were. And so we can use this kind of as a baseline to compare, are things better or worse than the peak at July 6th? And then we also do translating to the zip code level. I'll talk a little bit more about that. And then we're working actively to actually use this we've been forecasting using this because of this very strong relationship. We actually try to predict positivity rate, and eventually the goal is to kind of have a prediction of prevalence within communities in Houston. So in addition to kind of the Houston-wide graphs I showed, we also performed this correlation analysis for each individual wastewater treatment plant, where we compare the wastewater viral load, shown in green here, to the positivity rate for individual sewer sheds. And what you can see here is just, even across these sewer sheds that vary significantly in size. So really larger sewer sheds that serve 300,000 people versus ones that serve, 17,000. We see a pretty strong correlation between the wastewater data and the positivity rate data, again, giving us confidence in this approach. And then finally, we've also looked at kind of the total viral load versus the total number of cases in each of these sewer sheds. So here I'm showing each one of these dots represents a wastewater treatment plant. And the only difference between the green and the red is the green are the ones we've been monitoring since April, May, end of April, early May. And the red ones are the ones that we started monitoring a bit later. I think it was in June. But here we're basically adding up the total viral load that we've measured over time at these treatment plants and comparing it to the total number of cases in that sewer shed. And what we see is this really strong relationship or correlation between total viral load and total cases in the sewer sheds. Okay, so the way this information, so all of that was basically to show why we feel confident in using this wastewater-based approach across these 39 sewer sheds in Houston, and why the health department feels like this is a really wonderful tool to complement the clinical data that they get. And so the way this information is translated every week and communicated to health department officials is through plots such like this. So the first one on the left is showing the relative viral loads as compared to that July 6th date. So like I said, the peak date. And so here we're basically saying, okay, how does the current viral load compare to that July 6th peak? And then on the right, we're showing the percent 1 week change in viral load. So here it's looking basically like week over week. Are we seeing an increase or decrease in viral load at each of these treatment plants? And this is used to basically identify, okay, where are things getting better? Where are things getting worse within or across each of these 39 treatment plants? And so because we observe such a strong correlation between the wastewater viral load and the positivity rate, what we wanted to do next was then to use the wastewater viral load information to actually be able to predict positivity rates. So here I'm showing a predictive model. So this model was built using all the longitudinal data that we've collected, and it basically predicts the positivity rate using the current and 2 previous weeks viral load data at each wastewater treatment plant. And then it forecasts kind of the 2 following weeks positivity rate. So actually it forecasts the viral load in the treatment plant, which then is used to predict the positivity rate in that treatment plant. So here is kind of an example. So this is the actual positivity rate data shown in the black or the gray dots. And it's, somewhat messy in that it's variable. And then the blue line is the model predicted just from the wastewater viral load data, the predicted positivity rate just using wastewater data, and then forecasted 2 weeks out. And so this can be very powerful because it can basically be used to identify, again, like where are things getting better or worse across the city. And so now it's being translated out into at the zip code level. Because most people don't know what wastewater treatment plant service area that they live in, and all interventions are actually applied at the zip code level, the output at the wastewater treatment plant level is actually translated to the zip code level. And so many zip codes actually span several wastewater treatment plants, so that has to be kind of accounted for when we generate this. And then we, again, the wastewater viral loads in the purplish line and the positivity rates in the blue, and then the forecast is in these orange dots here. And this is, again, used to prioritize interventions at the zip code level. And the health department uses the wastewater data in addition to a lot of other data like clinical test data, vaccination rate data, And they generate these cluster reports every 2 weeks. And this is used to prioritize the top 15 zip codes um, and prioritize where interventions will occur. So this is specifically things like going door to door. At the beginning of the pandemic, they worked with community organizations and neighborhood organizations to actually go door to door and go to specific apartment buildings and tell them, where's the closest testing center, what are best practices to prevent the spread. Houston Health Department has several of these mobile testing units, so they could basically park one of these in a specific high priority zip code area. And then now we're increasingly working with health department to think about how to use this to inform vaccine rollout in these high priority zip code areas. Okay, now I'm just going to shift and briefly talk about how it's used at the not just the wastewater treatment plants, but also all these manhole and congregate living facilities and schools samples that we've been collecting. So these manholes don't have flow information. So we can't calculate the viral load in the same way, or we haven't been calculating the viral load in the same way we have with the wastewater treatment plants. But instead, we're just looking at raw concentrations. And essentially what we do is we designate sites as high, positive, lower positive, unclear, or negative. And then we communicate those levels to these facilities or these institutions that we're monitoring. And it's being used to basically identify spikes or significant increases in wastewater viral load at each of these different facilities. So here is an example of the manhole report that's generated. It shows kind of like all the different schools that we've been monitoring, the nursing homes, the jail shelters, and we specify the number of weeks it's been tested in a row in which it's had a high positive, and whether or not it's what the level is at that specific location each week. So now I think we're up to like the number of sites we're monitoring is increased even considerably since this report. And we've expanded to a number of additional congregate living facilities and also lift stations. And so this last slide is just showing the different lift stations that we're now monitoring as well via basically putting samplers at these lift stations to break up these larger sewer sheds into smaller sub-sewer sheds. And this is really a huge effort ongoing that the health department is working with Houston Water to basically identify locations, install these samplers, and then collect these samples on a weekly basis so that we get this more refined picture of how things are changing across the city. The most recent thing that we've been working on is to try and do variant detection and actually identify some of these highly transmissible variants of concern in our communities. So one thing we've been working on recently is using an assay that detects two characteristic mutations present on the UK variant. And so here on the right, I'm showing a table of all the characteristic mutations for this B117 lineage variant. And the assay we use detects two of these mutations, the 6970 deletion and this N501Y. And when both of these are present on the same viral genome, it's almost certainly a confirmation of the presence of this variant in the community. The issue with wastewater is that it's a mixture of many, many different variants altogether. And so it's really hard to have confidence that these two mutations are present on the exact same original genome. That being said, here I'm showing basically the presence, I'm showing the percentage of the SARS CoV-2 that we detected that has each of these mutations. So on the left, I'm showing this first mutation, the del 6970, and on the right, I'm showing this N501 mutation. And what's very clear is that since early February to where we are now in mid-March, the presence of these mutations has increased across to spread to basically every single wastewater treatment plant that we're monitoring, almost every single one, and increase in abundance. And the increase in abundance has been pretty comparable across these two different mutations. And this is really aligned with what we're starting to see in the clinical data, in which there's been a huge increase in detection in the UK variant in Houston. So, yeah, if anyone from Houston on this call is probably aware of this, but definitely be careful. I've been worried about this because these are obviously much more transmissible. And this is concerning that because while we see the viral load decreasing, we're seeing the emergence and increase in the presence of these highly transmissible variants. Okay, the very last thing I just want to mention is that we've been also doing some studies to understand the variables that impact the viral signal in wastewater. So, one of the things that people would love to be able to do is to be able to go from a concentration of a virus present in a wastewater sample and kind of actually be able to predict or at least have some insight to the number of people actually infected. Turns out this is extremely hard and you can't really do this because of so many different things that impact your ability to go from a concentration of virus to the actual number of infected people. And this has to do with things like variability in fecal shedding rates, duration of shedding, variability in wastewater generation rates, differences between symptomatic and asymptomatic individuals, and then also just differences in our sewer systems, right? So especially across Houston, we see some sewer systems that serve, cover like half of the city and some that cover very small geographic areas. And so decay and dilution are going to be different depending on the nature of the sewer system. So what we've been working on is doing some hydraulic modeling to look at the decay or the potential loss due to decay of the virus in the sewer system. So we use these hydraulic models to look at 10 different sewer sheds across Houston and to estimate the travel time or the amount of time the sewage spends in the sewer system before we grab that sample at the front of the wastewater treatment plant. So here I'm just showing under dry and wet weather conditions box plots of the travel times for these 10 different wastewater treatment plants. And while we see overall there's a little bit of a lower travel times for wet conditions, so we're using dry conditions to be more conservative, there's not actually a huge amount of difference between dry and wet conditions in the vast majority of these treatment facilities. So we use that travel time information and then we pair it with viral degradation rates. And so there's actually only a handful of studies that have been published that show decay rates of, that actually experimentally measured decay rates of SARS CoV-2. And what we also did some experiments in our own lab to measure it, and then we pulled from the existing studies that have been published, and there's really just not that many. But what you can see is it's pretty variable. It's ranging from 0.09 1/day to like 4.32, and obviously the temperatures are different. But you'll see even when we adjust for temperature, there's a huge amount of variability in the reported decay rates, really pointing to the need to have better, more comprehensive, more in-depth studies looking at the factors that impact decay of this virus in wastewater. So for example, if you pull kind of the lowest decay rate reported and compare it to the highest decay rate reported, most of the travel times in Houston are at or below around 500 minutes. But depending on which decay rate you use, and this is basically temperature adjusted to be 25 degrees in the dark lines and the bands represent 20 to 30 degrees, we're going to have very different findings depending on which decay rate you assume is correct in your model, right? Where half of the virus remains if you assume this faster decay rate versus virtually all of it's remaining depending on if you use this lower decay rate. So we basically took those decay rates and used that with the travel time information and then plotted these heat maps looking at percent virus remaining as a function of kind of where the virus signal would be generated from. And you can see, again, there's just a huge amount of variability depending on the decay rate that you use. So here I'm showing Chocolate Bayou, which is a smaller sewer shed in Houston. We can contrast that to 69th Street, which is the largest sewer shed in Houston. And with 69th Street, even with some of these faster decay rates, we see that a significant portion of the viral signal is potentially lost just due to decay traveling from these further, more distal communities to the wastewater treatment plant where we're picking up the signal. And so what you can use this information for is to actually think about where you might place additional samplers within the sewer shed, like we're doing with the lift stations, to break this up into sub-sewer sheds and just reduce the overall travel time and loss of signal due to decay. And we think that this approach of we think that decay is playing a potentially significant role because we basically compared the relationship between the number of positive cases in 69th Street to the viral load. And then we took those positive cases, and we actually have the health department actually has the locations of these. And so they were able to give us basically the travel time associated with each positive case. And if you basically correct or multiply through each individual positive case by the travel time, and then you look at the relationship between the number of positive cases corrected for by travel time and the viral load, we see that there's this improvement in the correlation between these two things, the wastewater and the positive cases. And so we've done this type of analysis for all the 10 sewer sheds, and we see that this accounting for travel time does improve this correlation, suggesting that decay is likely playing a role in many of the sewer sheds that we're monitoring. And so with that, I'm just going to conclude by, and I'm happy to take any questions, but this has really been an exciting and rewarding project to work on in collaboration with the health department, Houston Water, and Baylor. And the success of it has really resulted in the expansion of this monitoring program and the investment from our city to think about using this for monitoring other viral diseases and other pathogens of concern in the future. We spent a lot of time evaluating our methods, developing these statistical models, and really producing actionable data that's really used every week by our health department to inform interventions. We continue to iterate and improve and expand, and this is a kind of a learning process that never gets old. And so with that, I'd be happy to take any questions. Thank you very much.
Thank you so much, Dr. Stadler. We really appreciate you sitting down with us today. We are going to open it up for questions now. If you have a question, just type it in the left-hand side of your screen where it says attendee chat. And Dr. Stadler, you can just go look at those questions and answer at your own pace. I already see a few.
Sure. Oh, okay.
Starting with Dr. Naughton.
Okay. Oh, sorry about that. Okay. Do you see differences in the strength of correlation between wastewater cases for small versus large treatment plants? Yeah, that's a great question. So it's something we definitely need to look into more. There's kind of I would say almost like a sweet spot in the middle. So with the small treatment plants, the positivity rate data is super variable and sparse. And so the correlations are often really not great. And I don't think it's because it's not useful for small treatment plants, just we just don't have good case data at that level. Then at the really large treatment plants like 69th Street, I think we're running at the other side related to kind of this issue of potential decay and the fact that we're maybe losing some of the signal from the distal communities. And so we see sometimes slightly lower correlations with the case data at the really large treatment plants. And so, I think there's like some sweet spot in the middle where we have like enough solid positivity rate data to relate to the wastewater viral load, but we don't see a huge amount of loss due to losses in the sewer shed. Okay, next question was, which statistical models did you choose? Do you use a fecal indicator? So we don't use a fecal indicator. We just use viral load. So we, in that, like the way that we account for differences in sizes of treatment plants is largely just by multiplying by that flow rate. So every time we take we have a viral concentration we measure, we multiply it by the flow rate over the 24 hours that that sample was collected, and we compare the viral load. And in terms of the statistical models, they're almost all splines. So this is really a question for Kathy Enzer, but like the data is all fitted with these nonlinear regression curves that have some degree of flexibility. And so splines is what we've been using to both fit the wastewater data and fit the positivity rate data, and then do a Spearman correlation between the two of them. Okay, next question was from Andy Shaw. The current focus is on COVID. What is required to measure other illnesses? Any plans to do this? That's a great question. And yeah, we are planning and we're working on expanding this to look for other pathogens and viruses of concern. Many of the methods to measure biological contaminants, so things like antibiotic resistance bacteria or other viruses, are very, very similar. And it's actually just a matter of adding another quantification step. I mean, it's not as simple as that, but it can be as simple as just adding another target in the quantification step that will allow you to get more information on other viruses and other pathogens of concern. And so we've started to kind of think about what that list would look like in the first next iteration of this. And then we've been working with our health department to kind of prioritize which targets to go after, and then we'll work on the methods associated with that after that. But I am very much looking forward to thinking about how we'll use this to monitor for other things. Okay. Colleen asks, is there a plan for a public dashboard of the data? Yes. Oh my gosh, I feel like we're so far behind on this compared to everybody else working in the space. We have a dashboard drafted, working on making it live. It's just, I think we've just been inundated with all the other things we've been working on that it's gotten lower prioritization. That being said, like, we've really focused on communicating internally to our health department as like our primary user of the data, as opposed to the public, which, not that we're trying to keep this, away from the public. It's just we've been prioritizing the communication to the health department over the public dashboard, but we do have a draft of the public dashboard. We're just doing kind of the final touches on it before we turn it over and make it live. Okay, Paul Shu asks, this virus is a moving target, and you have moved past the time-honored bucket and rope gathering method. Given the decay in travel time, would you posit that large systems do need to establish monitoring stations to be effective? So I don't think that you can't still get information from monitoring these large sewer systems, and many wastewater treatment plants across the country and world are large, are at the size of 69th Street here in Houston, if not larger, and have still demonstrated these really strong correlations between case data and viral loads. I do think there's a lot we don't know about in terms of the form of the virus and the degree of decay that's occurring in the sewer sheds, and all of the different factors that impact the decay of the virus in the sewer sheds. So temperature, other constituents, other things about the strength of the wastewater that might impact decay. And to me, when we did this hydraulic modeling study, the variability in the data that does exist that's measured decay rates is just like shocking to be about how variable it is and how much we really don't know, and like how two different groups can come up with two very, very different numbers. And so to me, this just points to a knowledge gap and a need for our research community to do a better job at characterizing, so we really understand kind of how you compare across sewer sheds that vary drastically in size. Okay, are there plans to look for multiple variants? Can they be differentiated using qPCR? Jack Smith asked this. So yes, we are now actually doing like sequencing of the whole genome, to identify all potential characteristic mutations that might be indicative of variants circulating in the population. And the goal there is to combine a targeted approach where we use something like qPCR, ddPCR primers and probes to get really sensitive detection of specific mutations. So that's what we've been doing with the UK variant, but combine that with a sequencing approach that allows us to get a more kind of like general comprehensive picture of all potential variants that are emerging across the city. That's going to be less sensitive overall than kind of a more targeted qPCR based approach. So we're taking a dual approach. And actually, the way that we're doing it now is we're sequencing the wastewater all across the city. And then the health department uses that information to then go into individual communities. So let's say we detect specific characteristic mutations in one sewer shed. They're then directing like mobile testing units or contacting the neighborhoods that are doing the test, the facilities that are doing the testing in those neighborhoods, getting the PCR positives, and then sequencing those specific swabs to see if we can get confirmation, because it's much easier to get confirmation of variants from clinical samples than wastewater samples. But they're using our wastewater data basically to decide where to try and get those samples from, if that makes sense. Okay. Colleen asks, how do you manage to process so many samples? Seems like an overwhelming amount. Yeah, I feel overwhelmed on a regular basis. I have a really great group, and we have. So when this first started, it was like my grad students just like went all in, and everybody threw and helped out. And now I've hired a lot of research techs and a research scientist and a postdoc who've helped make this operational and like more manageable, because we do handle something like 200 plus samples a week in replicate. So we've been doing a duplicate analysis that ends up being like 400 samples processed per week. And we've invested a lot in the equipment that allows us to do higher throughput. And sometimes I do feel like we operate a little bit, like a commercial laboratory, which is not my intention at all. But so much of the stuff we're learning in real time. So we are doing like research at the same time we're processing all these samples. The end goal, and we're working really closely with the health department labs, is to basically hand this all over to them. So we will develop the methods and do kind of the research and the development on the methods, but then eventually the goal is that they're going to actually do all the sample processing week to week. We've just been doing it right now because they've been spinning up and getting the methods going in their labs. But once they do, I'm excited to hand over some of the work to them.
Okay, now there's two more questions, and they're actually in a different spot. There's an icon up top, a little chat box icon. There's two more questions in there that I don't think we got to yet.
Oh, here we go. Okay, can you repeat the reason for concentrating the virus before conducting the PCR test, especially if the direct extraction yields the higher value? Yeah, that's a great question. So the issue is that the direct extraction method is just not sensitive enough. So when you're only starting with an input volume of like 300 microliters of wastewater, the chance of being able to detect the virus in this tiny input volume is really low, right? Whereas with a concentration-based method, we're starting with an input volume of like 50 microliters, or 50 milliliters of wastewater, right? So several-fold higher volume going into it. And so our limit of detection and sensitivity is much better because we're starting with a much larger volume of wastewater being input into the method, if that makes sense. So while direct extraction had the highest yields, it was all of those measurements were at or below the limit of detection. I don't know if that's clear enough. So there's like the plot I showed was like back calculating to the original concentration per liter of wastewater, but we also have a plot in that paper that shows the raw concentration of the virus and measured in the actual extract that went into the PCR test. So that actual concentration in the raw extract is super low for direct extraction, to the point where it's like at or below that LOD, right? Whereas the concentration in the methods with the concentration is much higher and above that LOD reliably. Could this be used at events like spring break locations to indicate if they're truly going to be super spreaders? Yeah, I think it definitely could be. I think this is kind of goes along with the dorm monitoring that people have been doing. I think it's a good way to detect an outbreak relatively early and more efficient and more cost effective than individually testing people. And I think the most beautiful part of wastewater is that like there's no opting in or opting out. I guess you could not go to the bathroom, but like everybody goes to the bathroom, and therefore we have a sample from them, and it's anonymous, right? Whereas like if you want to individually test everybody at spring break, you'd have to like incentivize people with free alcohol or something like that. Okay.
Yeah,
I don't know if we're way over time, but do you want me to answer the one more in the chat?
Yeah, please. Thank you.
So the question is, what kind of technology did we obtain for this project? I think this is nanopore. Oh, so it's high-throughput sequencing. So we have been sequencing, we've done like many different approaches for the sequencing. Almost all has been Illumina-based sequencing. So the health department has a MySeq, and they've been doing using the Arctic primer set, if you're familiar with that, to do the whole genome amplification of SARS CoV-2 and then sequence it on the MySeq. We have a nanopore in my lab. We're going to actually test that out, I think, this week. We're going to sequence just the S gene, the spike gene of the SARS CoV-2 on the nanopore, but we have not done that yet.
Great. Thank you so much. We really appreciate it. I think we're going to wrap up today. I don't see any more questions. But if you can think of additional questions, you can email Dr. Stadler. Here's her email on the screen. And if anyone would like more information on becoming more involved with AAEES, please visit our website. It's aaees.org. You can always email me. My email address is mwaterman@aaees.org. And our next webinar will be on April 14th. Tom Ramsey from Geosyntech will be talking about ethics and engineering, so you won't want to miss that. Registration for that will start next week, so look for that email. Thank you so much, everyone, and enjoy the rest of the day.